Hello, everyone. First, I'd like to thank the organizing committee for inviting me to join this wonderful conference meeting. And here we can exchange ideas and learn from each other. And hopefully, I have already watched all the presentation. They are all wonderful. I learned a lot. And next, introduce myself. I work in Nutrition Immunology Lab at the USDA Human Nutrition Center on Aging at Tufts University. And I'm also a faculty member at the Freeman School of Nutrition, Tufts University. And my specialty, of course, is a nutritional immunology. So today, I'm going to talk about the role of green tea in regulating immune system with a focus on the host defense against the pathogen infection. And here, let me just briefly go over this introduction about composition green tea, and then very brief description of immune system that will help us to discuss and understand those studies that shows how green tea consumption affect immune function and anti-infection properties. So as we all know, after you brewed a cup of tea, the water extractable material released into the tea, which will be ingested. And among these water extractable materials, about 1/3 of them are polyphenols, mainly catechins. And we all know, or at least majority of studies have shown the health benefit of tea are mainly attributed to these polyphenols, and a variety health benefit already reported, including those in immune health benefit, which will be the topic of my presentation. And here are 4 major catechins. Among them, the EGCG is the most abundant and also perhaps the most biologically active. Therefore, many studies used EGCG as a supplementation to address the issue for tea's health benefit. And just by the way, and the studies can use the whole food in the form of a brewed tea or the whole tea powder, or the extracted tea, which is enriched in the polyphenols. And the other way is just using the purified form of catechins. Here is an illustration for immune system. And immune system can be divided roughly into 2 arms. One is innate immune system and the other is adaptive. Innate immune system is basically composed of the physical barrier and the cells, and the cells can be endocytes, which can engulf and eat up the pathogen or foreigners. And some other cells, like natural killer cells, can directly attack targets to break down the integrity of the pathogen. And some soluble components like complement, also the part of innate immunity. And the other part arm is adaptive, also called acquired. And innate immunity was born with, is genetically determined, and adaptive immunity developed later after encountering of the pathogens. So the third type of cells basically is a component of innate immunity, but is in addition to eating up the pathogen, more importantly, these type of cells can process the pathogen and transfer the passenger information to the innate adaptive immunity cell members. the adaptive immunity is composed of the mainly two types of cells. One is T cells, one is B cells. They are responsible for cell-mediated or humoral immune response, respectively. And for the property of the reaction, acute immune cells respond quickly, but it's not specific. They can only roughly detect the pattern for virus, bacterial, fungus, but not specific strain of pathogen. And they don't have memory. And in contrast, acquired immunity or adaptive immunity started later, slowly, but they are more specific, they are more intensified, and more importantly, they can have a memory, which is the principle behind vaccination practice. And I want to use the SARS-CoV-2 virus to explain how the pathogen invade host cells and the host immune system respond to destroy and eliminate the pathogens. So this virus has a protein shell and RNA kernels. And this is epithelial cells, and it is a bunch of a protein, but the most important protein called the spike protein or S protein. This protein can recognize and bind to the ACE2 receptor on the host cells to gain the entry. After enter the cells, and the virus can utilize the RNA to replicate, and using the polymerase and the protease. And this is antigen presenting cell, mainly dendritic cells. It can recognize the pathogen and eat up pathogen, digest it, process the specific signal expressed to the T cells. And the T cell has mainly two functions. One is a helper function. The other is a cytotoxic function. The cytotoxic function just like directly attack infected cells to break the cells to make the virus released up. And the helper function is help B cells. So B cell can produce the antibody, and this antibody is a soluble factor. They can bind to the virus, and so they work coordinately to eliminate pathogens. So the green tea or green tea catechins has basically two aspect function in terms of affecting immune system. So one is a defense related function, because the green tea can help the host defend against the invading pathogen. And the other function is regulating function, which serves to maintain the homeostasis, to maintain the balance, and to avoid overreactive or misdirected immune response attacked against the cells. So in the next, I will focus on the anti-infection, anti-virus and bacterial activity of green tea. And then just in the last two slides, I will briefly go over the anti-autoimmune disorders. And first, I'd like to just review the human studies. I kind of go over the two most recent meta-analysis and systematic review. And the first one is the meta-analysis summarized 8 human studies, including 5 randomized controlled trials, RCT, and 3 prospective cohort study. You can see here in the divided into the 2 type of a human study. In RCT, basically green tea supplementation cut the risk of influenza infection by about 1/3. And in cohort study, the green tea supplementation or intake consumption cut the risk of influenza infection almost by half. And the authors of this meta-analysis further divided the studies based on the administration route of the green tea or green tea products. As you can see here, either taking capsule or drinking green tea, then two approaches can be considered ingestion. Also the gargling. So that means just brew the tea, gargling, and spit out. And as you can see, either taking capsule or drinking brewed tea can cut the risk of the influenza infection by about 46%, and identical between the two ingestion approach. And in contrast, the gargling can reduce the risk by about 30%, which is lower than the ingestion, which is understandable because the gargling doesn't have the green tea absorbed in the body and only work locally in the mouth mucosal system. But that also tells us they can also be effective. Perhaps most likely is through the direct antiviral effect, which we'll discuss in the next. And here is the other meta-analysis and a systematic review. And we can see they covered 10 studies, including the 6 RCT, 4 cohort study, but the outcome measurements not only influenza, but also include upper respiratory tract infection, and also one study even include black tea. And as you can see, in the prospective cohort study, the reduction of the infection rate is about 1/3, and for RCT, the reduction of the risk is about 20%. As you see, the number is not the same as the one we just discussed, but because this study include different population, has different outcome markers, and also the dose of the consumption are different. About 2/3 of the studies selected in this meta-analysis is overlapped from with the previous one, the first one. And the author of the second meta-analysis pulled out the 2 studies that used the multi-doses of green tea supplementation. And as you can see, they built a dose-dependent curve, which will confirm a causal effect of green tea consumption with decreased risk of respiratory infection. As you can see, is quite linear, which further validate the efficacy of green tea in anti-infection. And here is a cohort study published more recently, not included in the 2 meta-analysis. This study used cohort, basically the workers in 4 companies in Japan, and the authors ranked the participants based on their consumption level of the tea, and they're divided into 3 subgroups. As you can see, as the seldom drinker as a reference, right relative risk to 1, and then with the medium dose and higher dose, you can see 20%, about 40% reduction in influenza risk. And the author further divided all the participants into two subgroups based on whether or not they received the flu shot. And as you can see here, in those without vaccinated with the flu shot, the reduction is about 24% in the medium dose and 52% reduction in the higher dose. And in those received flu shot, basically no effect. This is also easy to understand because the flu shot vaccination is very overwhelming in inducing the protection, and in relatively the green tea effect will be masked by the strong vaccination effect. Now let's move on to the animal models. We know the human study has advantage but disadvantage because we cannot do the intentional infection in humans, and also the natural infection rate is so low, and it is hard to conduct a human study with a large sample size. And for animal studies, you can mimic using the appropriate pathogen, and not only you can induce almost 100% infection rate, also you can kill animal to collect all tissues to do more intensive and insightful investigation to find out mechanisms. So in this study, the animals were fed the polyphenol, green tea polyphenol EGCG for 2 weeks. And then this animal infected with the coronavirus. And at the same time, you can see that after 2 weeks, and this is they measured the viral load in lung, and this is a control without EGCG or green tea, and this is with the EGCG, this is with the green tea polyphenol, so you can see greatly reduced the viral amount in the lung. This is just time course to show with the time post-infection, the virus amount, the number of the virus in lungs increase with the time. And this body weight is not changed. And in the same study, the authors also run the in vitro experiment to see the different level of EGCG added to how they affect the virus-infected epithelial cells. They use the cell lines susceptible to coronavirus. This is a non-infected. You can see the smooth, there's a high power microscope. And this is a control with infection without EGCG. You can see the virus particle. It's very dense. And here with the increasing level EGCG, 1, 5, 10, 20 microgram per milliliter, you can see the particle of the virus decreased with increasing dose of EGCG. And here is the virus marker protein. They used the Western blot to see the dose-dependent decrease in the cells. And here is a statistical summary for the viral load in the cells. And in this in vitro study, the authors used the brewed tea instead of EGCG or catechins, and they just boiled water, put a couple gram tea leaves, and after about a minute or so, pass through the filter to get sterilized, and then did serial dilution, and then they incubate with the target cell line and added either wild type or native SARS-CoV-2 virus or the variants or mutated coronavirus. And as you can see, this the brewed tea dose dependently decreased the viral load in all the cell lines in both wild type and the variants. So this is has a significance clinically because the vaccination efficacy will be decreased with the variation of the virus strain. And with EGCG, it doesn't matter you're mutated or wild type, they can universally suppress growth of the coronavirus. And here we discuss a mechanism. So there's basically two mechanisms. One is direct effect, and the other is indirect by enhancing immune response. So here's the direct effect, because green tea or green tea catechin have a direct antiviral effect because the virus has a spike protein, and green tea, EGCG, or T other catechins can bind the S protein to block this S protein to bind the receptor on the host cells to avoid entry of the virus into the cells. And the other way to defend against the virus is to inhibit the enzyme that's responsible for virus replication. So in both approach, the green tea or green tea catechin can inhibit the virus growth and replication. This is a direct effect without depending on immune system. And here is indirect, and the EGCG or green tea can modulate immune function. The first is in the lung, and you can see this green tea can suppress inflammation, because inflammation is critical for the severity and even mortality of the virus infection. Oftentimes, the virus itself doesn't kill you, but the cytokine storm kills the host. And the other way to affect the immune system to help to clean the fight against the virus is to mobilize the defense-related immune function to kill and clear the virus. And the other mechanism is green tea ingested in the gut can regulate the growth of the microbiota. So it result in a condition of anti-inflammation. So this anti-inflammation condition can go through so-called gut-lung axis to regulate the lung condition. And also whether, because the lung also the respiratory system is a mucosal system, and there must be some microbiota present, whether green tea can also regulate microbiota in respiratory system is under investigation. And now let's move on to bacterial infection. So we know the bacteria has is divided by gram-negative, positive, and based on staining. And here in this study, the authors use the model to give injection of the EGCG is to the abdominal cavity, and then they inoculate also in the abdominal cavity with the bacterial different strain, and at different time, either 24 or 8 hours before infection, or at the same time of infection, or post-infection, 4 hours after infection. As you can see, the green tea addition decreased the bacterial load in abdominal cavity, and seems like 8 hours before infection, administration of EGCG is most effective in all the, basically in all the strain of the bacteria. And as we know, the inflammation increases severity and causes for mortality. So let's see whether EGCG can alleviate infection through affecting inflammatory status. So here, the authors measure TNF-alpha, IL-1 beta, and IL-6 all of them are pro-inflammatory cytokine. You can see the decreased with the EGCG, and also consistently looks like 8 hours before infection is most effective. And here the authors show the pathology in lungs and the inflammation in lung. This is an air sac without infection. It's very clear. And this is a infection. You can see a lot of stained, which represent the cellular or liquid infiltration. A lot of immune cell infiltrated. And with EGCG, you can see this much more improved, close to the normal condition, away from the severe control condition. And it appears also the 8 hours before infection is most prominent. And this is survival. And they set up an additional group of animal and infect and follow up for 7 days and then counter fatality. And you can see here these only 20% animals survived at the end of study, but the EGCG supplementation before and after infection both improved survival, but 8 hours before infection is more effective in both gram-negative and positive. And since we talk about innate immune cells, which include neutrophil and monocytes, both are phagocytes, they are responsible for eating up, destroy the pathogens. So the authors inject the EGCG into abdominal cavity and then measure the number of these two cells. You can see this EGCG induced the increase of both cells, which indicate or suggest this may be this reason EGCG caused suppression of bacterial growth, and in both abdominal cavity and also blood. So both cell number increased. And here, author to further test causal relationship. So they use the infection model and they delete using antibody to remove the neutrophil or monocyte to see whether EGCG's antibacterial effect still there. As you can see, this is control bacterial load, and this is EGCG added. This is a non-specific protein as control for antibody, and this is specific antibody to remove the neutrophil, and this is for monocyte. And you can see the bacterial load back to the untreated. That means the EGCG effect is abolished by deleting the neutrophil and monocyte, which proved the EGCG's antibacterial effect is mediated by the phagocytosis function of immune cells. And the other arm of the adaptive immune system, the cells, like we mentioned, cytotoxic T cell, which is a CD8. And in this study, they fed animals 4 weeks with the catechins and then measured the both number of the cytotoxic T cell and also activity level functionality markers. And the EGCG or catechins increased not only the number of these T cells and also increased the activity level of these T cells. And here are another mechanism components. One is inflammation. This is a non-infection inflammation using endotoxin-induced lung inflammation. You can see this is normal, then this is inflammation. This is added green tea catechins. And this is using some reagents to block green tea related signaling pathway to diminish the EGCG effect. As you can see, this is a EGCG effect was eliminated. And here they show the cellular inflammation, and this showed oxidative stress. This is a positive control. This is EGCG treated, and this is a elimination of EGCG effect. As you can see, these are two inflammatory cytokine and show the inflammation state, and consistently with the cellular inflammation state, and the EGCG greatly reduce the inflammation. And this is all proof. Oxidative stress, inflammation are two factors inhibited by EGCG that contribute to the anti-infection property. And here I will just use 1 minute briefly go over the beneficial effect of green tea on autoimmune disease. Autoimmune disease characterized by overactive, misdirected immune response attacking self tissues. There's no cure for this disease, and currently use a bunch of different medicines to control the disease condition. And however, this medicine often comes a significant side effect, and the nutritional intervention can be alternative and a complementary approach to help alleviate autoimmune disease. And because green tea has anti-inflammatory and immunomodulating and antioxidant properties, so they are used to treat and prevent autoimmune disease. So far, a lot of animal models have been developed to demonstrate such effect, including myself. I have done many studies to show the anti-autoimmune function of green tea. And so far, the model involves the inflammatory bowel disease, rheumatoid arthritis, type 1 diabetes, and multiple sclerosis. However, the human studies is very limited. And although the animal model can mimic to a certain extent to the human disease, but this only in part. There's great limitation, and also because the metabolism of the green tea and the efficacy and bioavailability are different between animal humans. And given all this and interpretation of the data and the speculation and extrapolation to the clinical conditions should be cautioned. And here I just used one experimental result to give a taste how this animal model has been conducted about 10 years ago. I conducted this study using the called the autoimmune encephalomyelitis, which is a rodent model for multiple sclerosis. Basically, you inoculate or immunize with autoantigen to induce autoimmune disease, and then the animal developed the ascending paralysis, and then start about 10 to 11 days, and then you record daily for disease score, and also use pathology to see the inflammation, and also to stay for demyelination, which is the destruction of central nervous system. So you can see here, I use different dose EGCG, and this is control, and this is a summary of the under curves area to indicate accumulated effect for EGCG. Dose dependently inhibit the disease symptom and also improve the pathology. This dark color is infiltrated immune cells. You can see this control, this EGCG treated much better than this control without EGCG. And again, the pink color is the tissue destruction. And you can see this control, and this is pinkish, and this is a bluish, means much improved in preventing the tissue damage. And by showing this, I conclude my presentation and hope you learned something. Thank you very much.